All right, today we're going to dive into a real breakthrough in neuromodulation. It's called non-invasive temporal interference stimulation. We'll be breaking down how this technology allows us to precisely target regions deep in the brain, all without a single incision, based on a fantastic review by Xiao and colleagues in ACS Nano. So, for decades, neuroscientists have been stuck on this one huge question. How do we get in there and modulate those deep brain structures? The ones that are critical for everything from memory to movement, with real precision, but without the risks that come with surgery. This has been a major hurdle in developing new neurotherapies. And this slide really lays out the problem perfectly. You see, on one hand, you've got deep brain stimulation, or DBS. It's incredibly precise. We're talking targeting areas as small as 1 or 2 cubic millimeters deep inside the brain. The catch? It requires neurosurgery. But then on the other hand, you have non-invasive methods, like transcranial electrical stimulation, or TES. Super safe, but the problem is, it doesn't penetrate very deep, and the electrical field is just diffuse and spread out. So we've had this big clinical gap. You could have precision, or you could have non-invasiveness, but you couldn't have both. And that is where this incredible innovation comes into play, temporal interference, or TI. It's a completely new approach designed to give us the best of both worlds. The core idea behind TI is just so clever. What you do is apply at least 2 different high-frequency electric fields from outside the head. Now, individually, these fields are oscillating way too fast for neurons to even notice them. But and here's the magic, right at the spot where they cross deep in the brain, they interfere with each other. This creates a brand new, low-frequency signal, kind of like a beat, and that is the signal the deep neurons can actually respond to. It lets us sneak the stimulation past the cortex and activate only the deep target. Okay, so how does this actually work? Let's break down the mechanism. We're going to look at the physics first, and then the really unique biological response that makes it all possible. So, the physics is all about a principle called superposition. You deliver 2 high-frequency currents through different sets of electrodes. Let's say one is at 2 kilohertz and the other is just slightly different at 2.01 kilohertz. Where they meet, they create this envelope or beat frequency that oscillates at the difference between those two. And in this case, that difference is 10 hertz, which is right in the sweet spot for neurons to respond to. Now for the biology, which is just as cool. This whole thing works because of 2 key properties of our own bodies. First, the skull actually lets high-frequency currents pass through much more easily than low-frequency ones, so the signal gets deep inside with very little loss. Second, and this is absolutely crucial, the membranes of our neurons act like a low-pass filter. This just means that the neurons near the surface in the cortex can't keep up with the fast oscillations. They just ignore them. This lets the currents pass right by without causing any unwanted stimulation on the surface. So let's just recap that, step by step. First, those high-frequency fields easily penetrate the skull. Second, the cortex on the surface pretty much ignores them. Third, that useful low-frequency envelope is only created right where the fields intersect, at the deep target. And finally, only the neurons at that deep target actually respond and get modulated. And this leads us to the most critical piece of the biological puzzle, something called active rectification. When that low-frequency envelope signal reaches the target deep in the brain, the voltage-gated ion channels in those neurons are able to process it. They essentially tune into that slow-changing amplitude and translate it into a real, biologically meaningful signal. That's what allows us to actually change neuronal excitability, but only in that very specific targeted zone. Okay, the theory sounds fantastic, right? But what does the actual evidence say? Let's take a look at how this all holds up, both in computer simulations and in real in vivo experiments. Wow. Okay, this comparison really says it all. On the top, you're seeing the simulated electric field for TI stimulation. Look at that, a tight, focused, concentrated point of activation, right where you want it. Now look at the bottom image. That's conventional stimulation, or TACS. See how it's just spread out all over the cortex? It's diffuse, with no deep focal point at all. This visual just perfectly highlights how much more selective TI really is. And this chart right here, this just puts hard numbers to what we just saw. It's showing us the percentage of brain tissue, or voxels, that get activated. For TIS, you can see that a huge percentage of the activation is happening right inside the target region of interest, with very little spillover. But for TCAS, the effect is all over the place, with tons of activation outside the intended target. It's clear proof of the focality we saw in the simulations. And here it is. This is the smoking gun, the definitive proof from a living human brain. What you're looking at are fMRI scans during TI stimulation, and you can see it plain as day. Localized activity right in the deep target, in this case, the hippocampus. But just as important is what you don't see. No meaningful activity in the cortex sitting on top of it. This is what's known as cortical sparing, and it's one of the holy grails of neuromodulation. It proves the technique works as designed. So, the biophysical principles are validated. We know it works. But getting this amazing technology from the lab and into the clinic reliably? Well, that brings us to a pretty significant computational challenge. And that challenge has a name. It's called the forward problem. See, every single person's head is different. Skull thickness, tissue properties, it all varies. And these small variations can change the path of the electric fields in unpredictable ways. So to get that pinpoint accuracy we need, we have to be able to model exactly how the fields are going to behave inside a specific person's head with a specific electrode setup. So how do you solve it? Well, the tool for the job is something called high-resolution finite element modeling, or FEM. As you can see here, an FEM is an incredibly detailed digital twin of an individual's head built from their own anatomical scans. By running simulations on these personalized models, we can accurately predict where the electric field will go. And that is the absolutely essential first step to designing customized stimulation plans that can hit deep brain targets with confidence and precision.